내가 제일 나은 거죠. 내가 제일 나은 거죠. 내가 제일 나은 거죠. 내가 제일 나은 거죠.
Okay, can everybody see this first slide? My internet is kind of going in and out, so I need to make sure you guys can see it first. Okay, so since everybody can see it, even though I cannot see it, so let's get started. Um, so today we're going to talk about part 2 of cellular respiration. Last time we did all the introductory work for this chapter, and this time we're actually going to go into the steps of the cellular respiration inside a cell. Okay, so let's see. So there are 4 major steps. So today we're going to talk about the first 3. Okay, and then on next Wednesday we'll finish this chapter up by talking about the last step. So first step, as you see here, cellular respiration is very complicated steps, involve many enzymes. All of them involve enzymes, proteins, and they have to even individual steps. For example, in glycolysis, there are many steps within one step, like glycolysis. So remember, last time we talked about it's broken down into many steps, so the energy can be released in a very tiny package, so that it'll be usable. If it's released all at once, it will overheat our cells, and we cannot use. We will just end up wasting it as heat. So we don't want to do that, right? After this chapter, you will understand. Hopefully, you understand that our body is extremely efficient in extracting energy from food. And because that makes sense, in the old days, food is not very abundant, right? Whatever you get to eat, you need to be able to extract as much possible from food as possible, so then you can last longer and eat less. So that's the whole goal. But these days it's different, because the food we have is just everywhere, is cheap. With your body's amazing way to extract energy, you end up overeating a lot of times. Okay, so the first step is glycolysis. Now, if you look at this word, it has glycol, right? Glucose. Lysis, you already learned about this. Um, uh, Remember in hydrolysis? So it's meaning splitting glucose apart, okay? Second step is, this is a biochemical pathway. It means that the product from glycolysis will become the substrate for the next step, which is pyruvate processing. And then the product from pyruvate processing, which is acetyl-CoA, will become the substrate, the beginning substrate of Krebs cycle. So then all the stuff from all the products from these 3 steps will fit into the ETC chain. Okay, so it goes like this. So let's talk about glycolysis first. Cellular respiration actually takes place in 2 major locations inside a cell. Okay, glycolysis is the only step that happen outside the nucleus in the cytoplasm, and the rest of the other 3 steps happen in the mitochondria. So like I said, if there's oxygen around, glycolysis will split glucose apart. So that's why it's called glucose splitting. Why do you think glucose need to be split as the first step of the whole thing? Any guess? Good. You guys have really good guess. Remember the next step after glycolysis all go into the mitochondria, right? So if glucose, if you remember, needs a special transporter to get inside the cell through the cell membrane. So there are many mitochondria inside a cell. The mitochondria obviously is a lot smaller than a cell. So if glucose still need a transporter to go inside a cell, you can imagine that it'll be way too big to go into mitochondria. So that's why you need to split it outside the mitochondria so it can get inside. Very good. Now, so this is in general what we're gonna focus on, okay? Let me change it to a pen. What you guys supposed to do during these whole 4 steps is to focus on, remember we start with glucose. So glucose has 6 carbon, right? 6 carbon by single covalent bond. 3, 4, 5, 6. So every step that we're going to talk about, we have to break a single bond. Okay? And every time we break, remember there is like electrons, single covalent bonds share electrons like this. So in every step that we're going to talk about is you need to focus on number 1, you need to keep track of the carbon. Each carbon will end up becoming CO2, and you also need to keep track of which bond you're breaking. And that way you can keep track of the carbon, okay? So for example, in this step, when we talk about glycolysis, we're going to break this bond right here to split the glucose in half. And when we break this bond, remember there's 2 electrons right here. Every time you break a bond, last time we talked about the free electrons between the bonds, between the 2 carbon, is going to be picked up by electron carrier, NAD plus, right? So that way, when it pick up the 2 electron, it will become NADH. So it's carrying the energy of the electron. And when you break a chemical bond, you will make some ATP. Remember, chemical bond has chemical energy. So when you break it, this energy is released. So it's exergonic, right? Delta G will be negative, electron captured by NAD plus, but the energy within the bond is going to use to make ATP most of the time. Okay, so let's take a look. Now here's the key thing in glycolysis. And also in each step, what you need to focus on is what goes in and what comes out. So the inputs and outputs. Okay, so that way you'll be better. So I don't care much about all the tiny steps within each step, but you need to know what goes in, what come out, and which bond are you breaking, and what are you losing any carbon and stuff like this. So in this particular discard. In this particular step in glycolysis, where did it go? Here. In this step in glycolysis, you will see very quickly that even though the overall equation for cellular respiration require energy, require oxygen. Why doesn't have anything? Require oxygen. Let's try it again. But in this step in glycolysis, oxygen is not there yet. Okay, remember that equation we see, the overall equation for cellular respiration is the overall equation. It add up, it's a summary of the all 4 steps together, okay? It's not just showing you glycolysis. But anyway, glycolysis does not require oxygen. And you will see it really quickly. You will see that based on the inputs and the outputs. Okay, let's take a look. So glycolysis happens right outside the mitochondria in the cytoplasm. Split 1 glucose, which is 6 carbon molecule, into 2 3 carbon molecule called pyruvate. Now there are 3 steps. So here's what I mean by input. So in glycolysis, your input is, of course, you put in glucose. You have NAD plus, and I didn't write another one down, is ADP, and then your input will convert into output here. So the NAD plus will convert into NADH, right? The glucose, 6 carbon convert into 2 pyruvate, which are 3 carbons. And then if you put another input here as ADP, then the ADP will become the ATP. So that's why you need to know for each step what goes in and what comes out. There are 3 phases in glycolysis. The first phase is you actually need to spend some ATP to phosphorylate, to put the phosphate on the ends of glucose like this. And when you do that, you will make the glucose very unstable. So then you can cut it in the next step. So this is the next step. You will have the glucose cut in half like this, right? So each half will look like one half like this, right? Will look like this. And it's called gap. So this is called the cleavage phase, cutting phase. And at the last step here, you start getting the energy back because this P here will transfer back onto the ATP and more. Okay, so you go from gap to pyruvate, and so you spend 2 ATP in the beginning, but you end up making 4. So in glycolysis, your net gain is 2 ATP, you gain 2 NADH, and you have 2 pyruvate. Now, in glycolysis, the way they make ATP is through an enzyme. Okay, it's through an enzyme, and it's called substrate level phosphorylation because it's called substrate because it requires an enzyme. So if you look at this, ADP is here, and you have the gap here, right? The gap is the one with the phosphate group. So then the enzyme just transfer this phosphate to ADP to make ATP. Now, one thing to keep in mind is this: substrate level phosphorylation. The word phosphorylation means what? To put phosphate onto ADP, right? But it's at the substrate level means that it's using an enzyme. So ATP is made by an enzyme by transferring a phosphate from one molecule to another. But this process is not fast. So every time you see substrate level phosphorylation, it will give you a tiny bit of ATP, like in this step in glycolysis. You only get 2 ATP. Compared to later stage in ETC, you get way more ATP really fast. Okay. We don't do this because we're not, see, we miss another game. Okay, look at this. So what do you think the answer is supposed to be? Okay, most of you vote for C. Good, C is correct. Now we don't get to do this either, so let's go on. What about this question? We're looking for what is not true about glycolysis. Good, B, because the end product of glycolysis is what? Remember? NADH, pyruvate, and ATP. There's no CO2 or water anywhere, right? Good, B is the answer. Let's see what you guys get. Ooh, you guys are good. E. Good. Remember the 2 electrons are captured by NADH and the chemical energy is converted into ATP. Very good. So now let's now talk about how is glycolysis regulated, okay? So it's through something called feedback inhibition. So it means that if you look at this example here, look at this example here. The product of this chemical reaction, will go back to the beginning of the pathway and shut the first enzyme off. Okay, so in that way, it will turn the whole chain reaction off. Can you see that? So that's what feedback inhibition means. And what it means in cellular respiration is this. When you already make a lot of product, for example, if you have a lot of ATP already, then the ATP, which is a product, will go back to the beginning of cellular respiration and shut the whole pathway off. That way, you don't waste energy to break down any more food than you have to. Okay, so one good example, a daily life example, is your thermostat. When you set your thermostat to what, 70 degree, when your house reaches 70 degree, it will automatically shut the thermostat off, right? So that's an example of the product, which is the temperature that you set to, will end up shutting the whole process off. That way you don't waste energy and keep heating your room when it's already warm enough, right? Now, the other thing I want to mention is, remember, the product is always going to be a molecule, right? A chemical. And it will shut off an enzyme, not another molecule like a chemical, but will be an enzyme that will shut off. Okay, just remember that. So let's try this question. Good. It's this first enzyme that will shut off. So this is the product. It will come back somehow and shut that off. So let's see how it shut it off. And you learned that in the beginning of the semester in the protein chapter. Remember we talked about how to shut off an enzyme, you have two ways to do it, right? One is competitive inhibition. The other way is allosteric inhibition. So in feedback inhibition, you're doing allosteric inhibition. So the active site, for example, the example I have here is this is the first enzyme, phosphofructokinase, first enzyme for glycolysis. So there's two sites. One is an active site that binds ATP. There's also a regulatory site that binds ATP. So let's take a look at this. So this is the active site where it works on the ATP and carry out its chemical reaction. But when ATP is too abundant, the product ATP will come back and bind also the regulatory site. Remember, in allosteric inhibition, when it binds to the regulatory site, it will change the shape of the active site. So then ATP cannot bind to the active site anymore. So there's no more chemical reaction happen. Okay, just remember that. So that is allosteric, and that's how feedback inhibition works. So far we just done glycolysis. Is there any question on glycolysis? And then next step, remember at the end of glycolysis, we get pyruvate, right? Glycolysis here just happened here. We get pyruvate. Now pyruvate becomes the substrate for the next step, which is pyruvate processing. Now this step exactly do what it says. It processes pyruvate. So pyruvate, it is inside, processing is inside the mitochondria. So if oxygen is around, pyruvate will go inside. If it's not around, then we're going to talk about on Wednesday, next Wednesday, about what will happen. So we didn't go into detail about the structure of mitochondria, but today we will. So mitochondria is two layers of membrane. The outside is smooth, okay, like the purple layer here is very smooth. The inside blue layer folds into what we call crista. Okay, now you see all these space between these folding structure is called matrix. See that matrix here is between all these folding membrane. Okay, now the one, so you need to know where the matrix is because that's where most of the um, reaction take place, and the inner membrane fold like this, that's where step 4 happen. And the other space that you need to pay attention to is the intermembrane space right here. So it's a tiny space between the blue and the purple membrane, the outer and the inner membrane. This is a very important space, and we're going to talk about on Wednesday too. So these are the parts of the mitochondria you need to remember: matrix, inner membrane, and intermembrane space. Okay. Now, the 2-pyruvate from glycolysis is going to the matrix of mitochondria, and it will combine. Now, here's the picture. Pyruvate, which is in cytoplasm, go inside the mitochondria. This is mitochondria. This is outer membrane. This is inner membrane here. So when it go inside, it will combine with coenzyme A to form acetyl-CoA. So let's look at another picture. It's better this way. So this is pyruvate. Go inside the mitochondria matrix. Now, this time the bond we're going to break is this bond here. Can you see when I break this bond, I release a molecule of CO2. That's right here. And when I break this bond, the 2 electrons are captured by NAD+. That's why I become NADH. However, when I break this bond this time, I don't get any ATP. Okay, so I replace this CO2 with the coenzyme A. Can you see how COCH3, this part, is exactly identical as this part? Except the top, now we don't have CO2, we have coenzyme A here. So when you do that, you have acetyl-CoA. So in the glycolysis step, you break 6 carbon into 3 carbons. In this step, you break 3 carbon into 2 carbons. So we only have 2 carbons left. So the input for pyruvate processing is, of course, pyruvate. You'll have NAD+, right? You have coenzyme A. So these three are your inputs. And what comes out? You have CO2 that comes out. You have NADH. And then, of course, you have acetyl-CoA that comes out. So these are your outputs. Good. B is the answer. Very good. So now after pyruvate processing is done, the product of pyruvate processing will go into the next step, which is the Krebs cycle, or it's called the citric acid cycle. And once again, this is in the mitochondrial matrix. So when pyruvate processing is done, the product can immediately go to this because they're in the same location. Now at this step here, glucose is completely oxidized. That means there's no more energy left in glucose. And let's focus on this part, okay? So acetyl-CoA, which is from pyruvate processing, will go into citric acid cycle. And each, remember this is 2 carbon now. Acetyl-CoA has 2 carbon. And the 2 carbons in here will be completely liberated as CO2. When it becomes CO2, there's no more carbon-carbon bond. That means all the carbon-carbon bond is broken. All the electron has been extracted. So there's no more energy left in glucose. That's what I mean by glucose is completely oxidized. Okay. Of course, the energy between the carbon-carbon bond turn NAD plus to NADH. And this is a cousin of NAD plus. They do the same thing. They're electron carrier. And so when this pick up electron, it becomes FADH2. And of course, the chemical energy can convert into ATP. Now, this step is just like in glycolysis. The way they make ATP is by substrate level phosphorylation. So it's using an enzyme transferring a phosphate from one substrate to another. So it's a very slow process that you make very little ATP, okay? Now here's what a citric acid cycle look like. So the input is the acetyl-CoA from pyruvate processing. When it goes into the cycle, it will combine with oxaloacetate here. So this is the molecule you combine with. So this is 4 carbon. The circle just telling you how many carbons. 4 carbons combined with the 2 carbons. Then you make citrate. That's why it's called citric acid cycle. It's a 6 carbon. 1, 2, 3, 4, 5, 6. 6 carbon molecule. Because you combine 2 carbon and 4 carbon here, it becomes 6 carbon. And they go through many steps. Then it liberates CO2, right? Generate NADH, generate ATP, and also some FADH2. and then it will go back to what it originally started as, which is the oxaloacetate, and then can combine with the new acetyl-CoA. So then this go on and on. That's why it's called a cycle. Whatever you use as the first step will get regenerate at the end of the cycle, so they can go on again. You can see that in this cycle here, a lot of CO2 is made, right? Because you liberate all of this carbon, so you break this carbon-carbon bond here. And you also get a lot of reduced electron carrier, NADH and FADH2. Now, if you look at what is the input for citric acid cycle, what goes in is acetyl-CoA. Since you're generating all these electron carriers, then it has to, so besides acetyl-CoA, you have NAD plus going in, FAD going in, and ADP going in, right? So what is the output? You have ATP, you have all these electron carriers that are reduced coming out, and of course the acetyl-CoA, the input acetyl-CoA becomes CO2. So just remember, every input needs to be accounted for at the end as an output. So this is the output of acetyl-CoA. So for every glucose, you have to do 2 turns of the Krebs cycle because you will generate 2 acetyl-CoA. Okay. So here is keep emphasizing that at this step, at the end of citric acid cycle, glucose is completely oxidized because there's no more carbon-carbon bond. Every single one of those carbon-carbon bond between within glucose are all broken. Okay, the first one is broken during what? Pyruvate processing, and then the rest is broken during this step here. So every time, like I said, when the carbon-carbon bond is broken, you liberate it as CO2, and then you liberate it as CO2, and then you get NADH, and then you get some ATP sometimes. Now, are we okay? So this is the 3 steps. And then we'll talk about the last steps and fermentation on Wednesday. The key word in this question is most. Where is most of the energy? Okay, let's see what you guys have. Okay, good. Most of you vote for B. Now, although A is a true statement by itself, but that's not where most of the energy is, because so far you only generated a few ATP, right? But you through the first 3 steps you generate a lot of NADH. Now, Krebs cycle and pyruvate processing is regulated the same way as glycolysis. That means the product can always go back to the beginning of the process to shut the enzyme off. So then the whole thing will get shut off. Now here's a summary if for those of you that are confused. Now here's how much ATP you made so far after the first 3 steps. You only make 4. Now remember, our bodies spend millions of ATP per second. So 4 ATP is next to nothing. However, 1 molecule glucose after the first 3 steps, you have 12 reduced electron carriers. That's a lot. And then, of course, you break down all the glucose. So the 6 carbon in glucose becomes the 6 carbon in CO2. Okay. So the catcher is this. Of course, the 4 ATP you make during these 3 steps can be immediately used, right, for whatever cellular work that needs to be done. But the 12 electron carriers will go as the substrate, will work as a substrate to go into the last step in ETC, the electron transport chain. And this is where all the energy is going to be turned into. So the energy in the electron in these guys will turn into ATP in a very, very quick and efficient way. So we're actually done. So this is a short part. I'm going to save some for the last part in the last step on Wednesday. So remember, Monday we don't have class because it's an exam day. So you need to finish exam 5 by 11:30 p.m. on Monday, next Monday, which is April 20th. Now, before I go on and talk, I will show you after this the option for biofuels. So I make a table, and then you guys need to, I'll post it on D2L. You guys need to think about which option you like, and then you start doing some research and thinking about your question and stuff, and we'll go over it again on Wednesday. Okay, so remember Monday, April 20th is the exam day, so it's no class, no live stream, but I will have a study session on Monday at 11:30 to 12:30 in case you have some last minute question for the exam or for whatever you want to ask. Same link. Okay, so study session that day, April 20th. And I'm going to pull up the option for, actually, let's talk about the worksheet first for today, in case you have questions. Let's look at the worksheet. I think most of this on the worksheet will be okay for you guys, except this part here. Number 8, question number 8. Now, this is one of those difficult true and false questions. Because if you say it's true, let's say the statement number 1. If you say it's true, you just write true right here. You don't have to do anything. If you say it's false, for example, this sentence here, what is wrong? Glycolysis happens in the nucleus. What is wrong with this sentence? So it's not a true statement. So what is wrong with this statement? Come on, people. What is wrong with this statement? And you need to underline the wrong part, and then you have to rewrite it so it'll be correct. Good. So nucleus is wrong. So what you're going to do, you're going to underline nucleus, and then you're going to write here in replace nucleus, you're going to write cytosol, right? Like that. Okay, so this is an example. Now, another one I want to give you an example is sometimes you just need to add some silly things. For example, here, C is this statement correct or incorrect? If it's incorrect, tell me how can you fix it to make this statement correct? Good, you guys all got it. So it's a false statement, and all you have to do is put not in here. You can underline the is and then change it to is not. Right? Are you getting the gist how to do this? This is exactly how you're going to do it. And this will help you to clarify some concepts. So then it's the same kind of true and false on for Krebs cycle. You should be able to do it. Now here's the part. You need to draw a concept map of glycolysis and pyruvate at the end. So you need to include well, where it happens, right? Location, the inputs, outputs, the phases. For example, in glycolysis, you have the 3 phases, what happened in each phase. So this will be a great summary of what you learned today and help you to kind of solidify the stuff from today. Okay. So this is the worksheet for today, and it's not due until Wednesday because Monday is exam day, so there's nothing due. This worksheet is due on Wednesday, April 22nd. So now let's look at the biofuel stuff. Option. Where's option? Oh, here. Now this is the options. We have 5 options for different biomass and the enzymes of choice. So what I'm going to post this on D2L, so you can take a look at it under today's material. So what you need to do is you need to research. Of course, you should have researched the enzymes in the design part, and you're also going to research some of these biomass to decide which one will give you the most alcohol with the combination of enzymes that we have here. So basically what you're trying to do is to pick out one option out of the 5 that you believe will give you the most alcohol. Okay, and I have the data from previous semester. And so once you pick one and you do your design part, you turn in your design and I look over it, make sure it's correct, and I'll give you those data, then you can do the report. So is that clear what you're supposed to do? Okay, cellular respiration is not on the exam. So for exam 5, it's going to be covering genetics, meiosis, and DNA replication. So it's three chapters. Okay. Cellular respiration will not have an exam, but it'll be in the final exams. So it will not have a separate exam on its own, but it will be included in the final exam. So is there any other question? Now remember, I'm going to post it on D2 on Monday. Even though there's no class at 11:30, we'll still have a study session. Okay, and then you can come and ask questions about the exam. Of course, before taking the exam, when you go over your study material, if you have questions, you can ask me about those materials and stuff like that. So there's not a particular due date for the different, there's only two parts for the biofuels lab. The first part is the design part, okay? I think it's the first 3 pages on the data sheet. If you get it done, you can turn it in, and then I'll give you the data for the option that you pick. And so the biofuels lab is still the end of the semester. So you have until what? The last day of semester is May 11th. So you have plenty of time. But I'll keep reminding you along the way, you need to get this done so you won't be cramping everything up. But the catch is this, you have to finish your design part before I will give you the data. If you never finish the design, then you don't get the data. So there's not a set due date. Just do it, send it to me, and I'll give the data to you, and you can send the report in. So the whole thing is still May 11th. Yes, Harley. So when you pick whatever option you pick, you just email me, say, Dr. Fan, I want option 1 or 2, whatever it is. Okay, so I'll write that down, and then, or actually, you don't even have to email me. Whatever option you pick, you just use that to do your design. And from reading your design, experimental design, I'll know what option you pick. Does that make sense? So you just pick the option you want now, do some research on it, and then we'll talk more about the design on Wednesday. But I encourage you, before you pick any one of these options, to do some Googling, right? To look at what does orange peel contain, potato skins, and to see whether the enzymes of choice make sense to you, whether that kind of biomass is something that will give you a lot of alcohol. So don't just randomly pick. Okay, any other questions? If you have no more questions, we're done for today. So you can log off if you do have questions. Of course, you can ask those questions now. Yes, and very good question. So when I have those, that means that group of students, they test different enzymes, see which one work better on that biomass to convert that into alcohol. And we'll talk more about, if you think this is a good option for you, we'll talk more about how you would set up the experiment with that option. Samantha, the exam is going to be covering DNA replication, meiosis, and genetics. Actually, I'm going to put this document into the biofuel folder. That way you don't get confused. Yeah. Today 17. Okay, since no one has any questions, I'm gonna end the live stream right now. Okay, if you can think of any, you can always email me, or I will see you on study session next week. Okay.